this system. From there onwards, we continue to post a month. We are having lectures, and particularly we are proud to say that we used to have lecture on October, 3 lectures on October on Nobel Prize. Every year, Nobel Prize is being announced from starting from first Monday up to the weekend, and the next week first Monday they are announcing the economics. We cover Nobel Prize on medicine, physics, and chemistry. Every year we successive 6-7 years we have done the lectures on Nobel Prize. It was an interesting lecture. All our lectures are in the YouTube channel, PSL YouTube channel, Popular Science Lecture YouTube channel. I'll appeal to you to make use of this opportunity to visit our PSL channel. I also request you to subscribe to the PSL YouTube channel.
No,
So before jumping into the lecture, let me introduce Tamil Nadu Science Forum. Tamil Nadu Science Forum is a voluntary science organization. It was started in 1985.
let me check
Few research scholars in Madras University, particularly from nuclear physics department, some 4-5 lecture scholars have joined together and founded the Tamil Nadu Science Forum. From then onwards, they used to conduct hall lectures in different places in Chennai, particularly in University of Madras, in both campuses. And at some point in time, they decided to go out of campus and go to the place where people are gathered, and we'll have a lecture at the streets and other places. They prepare slides in the slide projectors, and instead of this LCD projector, we used to have the films. So they prepare a lot of films and do the lectures in the streets, and it was going on for 5 years. And they decided to expand it to throughout Tamil Nadu. It was a science yatra was held in 1986 from Kanyakumari to Chennai. They went to the different district, different schools, different places and different villages, and they have the lectures. Based on that, it became a popular movement. And based on this popularity, the government of Tamil Nadu has given opportunity to work on the literacy program. So Tamil Nadu Science Forum was a nodal agency for pushing the literacy program. We involved in the literacy program in Pudukkottai district. Initially, it was a model district. They reached 100% literacy. Then we moved to Virudhunagar district. Finally, it extended to entire Tamil Nadu. Tamil Nadu Science Forum was very busy on the literacy program from 1989 onwards. Then 1989, we started a magazine called Tulir. It was a children's science magazine in Tamil, and the next 2 years, we started an English magazine called Jantar Mantar, is meant for the college students. So with that, the journey was continuing. When the literacy program was going on for 10 years, then we go for a post-literacy program. Finally, the organization has expanded to entire Tamil Nadu, and we have district body, we have the local branches as well. Now the state level body as well. The state level body is divided into 5 different work groups. We have 5 verticals and 5 work groups, 6 work groups. So one work group is working on science communication. Basically, what we are doing is the science communication. The next one is the science publication. We have 2 publications. One is the Jantar Mantar and Tulir. Apart from that, we are having a lot of translation books in Tamil. That is they're taken care by science publication. The third one is we are working on the development and environment. So we need development simultaneously. We need to protect the environment as well. So how to balance these 2 things? Some of us are working on that, and we also work on health and education. And another area is we want to bring more women in science, for which we have a separate body called the Samam. Samam is working on bringing more women on science. And again, we are focusing on youth. There are 6 verticals, we are working on it. That's an interesting thing. And I would like to make an appeal through this forum to invite you to become a member of Tamil Nadu Science Forum. We have this PSL group is part of the science communication. We feel that in science communication, we do a lot of things in schools, but we have not done many things for the colleges. Then we started the PSL program. It is basically focusing on higher science, the latest research and other things. So we come to the 85th lecture. We have a Telegram group, we have WhatsApp group. Apart from that, we have the Google group. If you leave your email ID in Google group, we will add you in the Google group. We have a QR code for becoming the member of Tamil Nadu Science Forum. We have the QR code for the feedback form. If you fill up the feedback form, we'll pick up your email ID and add it in the Google group. So we will be displaying the QR code for the WhatsApp group as well as the Telegram group. You can become a member of that. Coming to the lecture, so I'd like to introduce Dr. Sayantan Datta. He has written a lot of articles. I want to list out the articles, but he says that no, leave it to me. I will do it. He's going to add it in a slide. He has written a lot of articles. In 1980s, when I read when I was doing my engineering study, I heard that the computer virus. What does the virus do with a computer? Virus is a living organism and computer is a non-living thing. How do they get connected? I was very much wondering. Then I find out, then when we go through the classes and find out what is computer virus. Now, the bacteria is going to be a computer. That's what he is going to talk today. It's going to be an interesting lecture. I invite Sayantan Datta to deliver it.
I have that. Am I audible? If you can't hear me, you must tell me that you are unable to hear me. I might. So, can you hear me? Okay. Oh, perfect. Yes, this is good, right? All right. My name is Sayantan. I come from Krea University. I teach at the Center for Writing and Pedagogy. I'm trained in neuroscience originally, but I quit a PhD in neuroscience to do science journalism. And since I did not finish my PhD, since that time, I've been writing for several national and international publications on various aspects of science, of which gender and science is one of the important areas that I'm focused on. Because I have some training in neuroscience, and I initially set out to be a neuroscientist studying brains. One of the things that I report on quite actively are new innovations and discoveries in neuroscience, of which intelligence particularly is something that has stuck on. Sorry, I don't have a list of my articles on the screen. I must disappoint you, but they are not hard to find. If you Google my name, I believe it should be there. I'm not boasting, just suggesting. Okay. So yeah, what I'll be talking about today is intelligent behavior in brainless organisms. Towards the end, I'm going to talk a little bit about what Dr. Vijayan was talking about, which is how bacteria are being engineered to reach some kind of computational status, but some groundwork before that, perhaps. So why are we talking about intelligence? Does the word intelligence ring anything to you in today's context? Yeah, artificial intelligence. It's almost unavoidable if you step out into the world. And that's interesting because I don't know if you're familiar with this image. Does anybody know this image? Does it remind you of something? Okay, this is called the Great Chain of Being. This is a picture of the Great Chain of Being from somebody called Didacus Veladus. This is 1579, so the medieval times. And this is how the world was conceived in the medieval times, right? So you had God at the top, and God, along with his angels, were in this realm, and they were sort of omnipotent, omnipresent, and omniscient. So they could do everything, they were everywhere, and they knew everything. Below the God were human beings who were cast in a somewhat poor image of God, but yet came right after because they were reasonable. And reason is the first mention of intelligence that we have in history. Aristotle and Plato used the word reason to talk about man. Below the humans were animals, then plants, and then minerals. And at the end of minerals, you have hell, and we don't have a lot of information on what's happening there. So this is how, for a very long time, the world was conceived. And reason was one of the more critical parts of humanity finding a slightly elevated status in this imagination, medieval imagination of the world. The reason there is very important. Let me now bring you to 2024 in the month of November. This is news from 2 days back. This is the Peters Chapel in Sweden, and something very exciting has happened there. The green light you see, you're familiar with this architecture. If you've been inside a church, you probably know of a confession box. Do you know what is a confession box? If you've committed any sin, you can go inside the confession box. There is a priest sitting on the other side, and you can confess to the priest, and the priest will give you some penance, right? So write 100 Hail Marys. That's one penance. What these people have done is that they have replaced the priest in the confession box with an AI. What do you think the AI is occupying the role of? Very interesting. It's actually not the priest, it's an AI Jesus. Yep, they have a very interesting collaboration with a tech university nearby, and they were deliberating on whether they should get an AI priest. And then they decided, why get AI priest when you can get AI Jesus? So they call this installation the Doi in Machina, which means God in the machine. And people have come, thousands of people have come from different faiths, from different parts of the world to interact with our AI Jesus. And an astoundingly large number, almost 2/3, I believe, has said that they had a religiously positive moment with the AI Jesus. And the pastor of the church has been quite surprised that that happened. So this is an AI machine that was trained on theological texts, and then you could go and tell this machine life's problems, and this machine could respond. And people had a great Jesus moment with our AI Jesus. So we are actually at a time when artificial intelligence is quite literally replacing God in God's own institutions. And that warrants us to talk about intelligence in some depth today. That is what I will be trying to do. And that's where we must begin, which is, what is intelligence? Anybody wants to take a crack on defining what is intelligence for me? I have indefinite patience, so I'm very happy to wait.
Adaptability.
Oh, sorry?
Adaptation.
Adaptability, right? So something around you changed, and we need to continue surviving, and as a result of which we make changes to ourselves. That is adaptability, and that is certainly a great defining feature of intelligence. Anything else? Analytical ability, right? So if you're given a problem to solve, you should be able to follow a series of steps to solve the problem. Okay, you had your hand up. Sorry? Okay, one of the things that we talk about complexity of organisms is that you show reflexes, right? So a stimulus evokes something quickly in you. Absolutely. Anything else?
Learning ability.
I beg your pardon?
Learning.
Learning, absolutely. Intelligence is most commonsensically associated with learning. If your children or people who you don't know are not learning well in school, schools will often say you're not intelligent. That's not the way biologists talk about intelligence, but learning is an important component of intelligence, absolutely.
You see, the impact of the
Reading, listening, and listening, and
Okay, that is an important way of defining learning, that it is actually acquiring, it is responding to stimulus, experience, responding to experience, remembering it, which is memory, and then using it in a different context. Absolutely. Creativity and innovation. Absolutely. That is, sorry? Absolutely. Design thinking, creativity, innovation, all important aspects of intelligence. Thank you. You've pretty much done my job for me, but because I have my slides, I must. Our story of intelligence starts with the great granddaddy of philosophy, Mr. Plato, who said that reason must rule over spiritedness and passion. So for the Greek philosophers like Plato and Plato's disciple Aristotle, not only was reason, or what we think is the closest equivalent of intelligence in antiquity, not only was it a characteristic of human beings, defined human beings as something else from everything else on the planet, but reason was also seen as something that should be used to curb emotions and passions. That was the thinking in antiquity. Now we have a fairly complex definition of intelligence. It varies from context to context. But since the 1900s, these are what different people have told us intelligence is. There's this man called Binet who gave the first IQ test. If somebody's from psychology here, they might know, but the IQ test was the first version of it was designed by this person called Binet. And he said that intelligence is a combination of judgment, so being able to make a decision between two alternative options, adapting to environment, which is something that Sir there told us, and autocritique. So autocritique means thinking about something that you've done and reasoning whether that was right or wrong. So that was, according to Binet, the three defining characteristics of intelligence. A little later, somebody called Wechsler, and the names are really not important, came, and this person said that if you are intelligent, you must act purposefully, which we now call goal-directed behavior. So you have a goal and you want to meet that goal, and your actions must demonstrate that. You must think rationally, which means there is logic and reason that's guiding your thought process. And you must adapt to the environment. So if there are changes in the environment, we must be changing ourselves, not really at an individual level, but at the larger level of a collective to adapt to that environment. Humphrey's definition was easier. It says intelligence involves learning, memory, and its use in new context. So you have experiences in life, your brain changes as a result of those experiences, and then you implement what you learned from those experiences in a different context. That defines intelligence. Somebody called Gardner, who's still alive and is a professor, I believe, at Harvard University. He's also called Howard Gardner. He said that intelligence is a combination of problem solving and problem finding. That is also important. He said that solving problems alone is not adequate to be intelligent. You must also be able to find new problems, solve. And several people spoke about goal-directed adaptive behavior, which basically means that there is a purpose, there is a goal, you need to get that goal. To get that goal, you're constantly modifying yourself, adapting to the environment to meet that goal. And finally, very recently, somebody gave us a formula, an equation for intelligence. I'm sorry, I don't completely understand this, but from what I understand, this person sees intelligence as a force that is dependent on something. I don't understand it. I don't think that this equation captures the complexity of intelligence for us, but yeah, I thought I should show you the picture. It is F is equal to T grad S tau, where F is the force of intelligence. This person sees intelligence as a force, and T is the strength of this force. I don't think strength is a physical term in physics. S is a series of possible events, and T is at a particular time horizon tau. That's all I know about this equation. I'm sorry, I can't talk more about it. I don't think it's particularly useful, but there is an equation. People have tried to mathematically capture intelligence. The problem with all of these definitions, although they are great, is that they do not give us a lot of things that you can study in the laboratory. If you're a biologist, you need quantities that can be measured, and those quantities have to be operationalized. So for example, if I ask you what is learning, different definitions can emerge. We need to agree on one definition, even if that is not the most accurate or the most real definition, because it allows us to measure, capture certain things. So biologists typically define intelligence as a combination of these characteristics. They say that learning and memory is certainly an important part of this, as is abstract reasoning. Everyone understands what is abstract reasoning? And abstract reasoning is when you take a series of examples and then you can say something about the concept. So let's say there is this chair, there is that chair, there is that blue chair, which is more comfortable. If I put all these three chairs, what is it that we can say about all chairs? That is abstract reasoning. To take, to draw upon examples to make a more general comment is an abstract reasoning process. Then there is something called self-awareness. Self-awareness actually has a very specific definition in biology, which is if I put a red bindi on your face, will you look at your mirror and recognize that you have a red bindi? That is the definition of self-awareness. People have tried this out with crows, for example, right? So you take a crow, you put a red dot on its head, and you see the crow watches itself in the mirror and tries to get rid of the red dot. If the crow does it, then we say that the crow is self-aware. The crow knows that the red dot is not supposed to be there. And crows do seem to be self-aware. Honeybees seem to be self-aware. You put paint on honeybees, you show them a mirror, honeybees will try to remove the paint. That is an act of self-awareness. Decision-making, problem-solving, creativity, all of these are seen as different properties of intelligence. And it's pretty much impossible to study all of these together. So people pick and choose what they want to study. I want to focus today on two of these. One is learning and memory, and the other is decision-making. Learning and memory because it is perhaps the most worked on in this entire list. Decision-making because that is where we have had some of the most interesting findings in terms of intelligence. We'll see how that goes. Okay, where intelligence? If I ask you, what is the seat of intelligence in the human body? What is the seat of intelligence in the human body? Brain. Obvious answer. The brain is the seat of intelligence in the human body. Interestingly, that was not always the case. Ancient Egyptians, for example, did not know what to do with the brain. Does anybody know what they decided to do with it? They decided to throw it in the dustbin. I'm not kidding. When people were mummified, their hearts, their kidneys, and their lungs were preserved along with the mummy. The one organ that was taken out the fastest and trashed was the brain. And that is because ancient Egyptians, like many other cultures at that time, did not know what the brain can do. They thought that the seat of the soul was the heart, so the heart needs to be preserved. Water was an important force in the body, and they had done dissections of kidneys, so they had seen urine being formed droplets of water in the kidneys. So they knew that the kidneys were important, and they knew that lungs help you breathe, and so lungs were important. The brain, they used to put a needle through the nose, pluck everything out, and trash it. It's only with this Greek physician, Greek Roman, perhaps, yeah, both, physician called Galen, or if you want to be very picky, you will read this as Galenus. This person, this was an important and interesting person. He fled from Greece to Rome and became one of the physicians to the Roman emperor. And his task was to work with gladiators. Everyone knows who are gladiators. There was a time in Roman Empire where people loved to put men to fight. And there was a big arena. These two men would fight. At the end, the winner would ask, can I kill this person or shall I kill this person? And the audience would collectively decide whether this person gets killed or not. Bad description, but accurate. What Galen observed is because gladiators are fighting a lot, some of them got injuries to the head, as a result of which they started behaving oddly. Some of them lost the ability to speak. Some of them started speaking nonsense. Some of their personalities became entirely different. So because of their injuries to the head. And from there, Galen concluded that the human body has 3 souls, because one wasn't enough. Galen said that the brain is the site for the rational soul, the soul that helps us make rational decisions in the world. The heart was the site of the spiritual soul, which is what keeps us connected to God. And the liver was the site of the appetitive soul, which is what keeps us eating. And for Galen, that seemed to describe the world fairly well. This changed substantially in the 1900s because of one invention, which is of the microscope. Once we had the microscope, we could peer deep into the brain. And anybody knows what we found? Yes, we found these squiggly little things called neurons. Neurons are nerve cells. They are one of the cell types that makes up the brain. There are other cell types, but this is perhaps the one that determines the function of the brain the most. And neurons, this is what it is called. And these neurons are actually very easy to distinguish. They have these branches on the side, and that part is called a cell body. Then there is this long tail called the axon, and this tail ends at these knobby things called boutons, synaptic boutons, right? And neurons are often connected to each other, and this connection is called a synapse. Everyone knows this, I think. It's okay. Yeah. What is interesting is neurons can conduct electricity. So they communicate through electricity. So you have, when I say electricity, I mean that the concentration of ions across 2 sides of the membrane is different, which creates an electric current on the membrane. Does that make sense? There are more positive ions on the outside, lesser on the inside. So you have a potential difference. The moment there is a potential difference, there will be a current. So there is a current. The neurons communicate through current. The current is generated somewhere here, and it moves through the axon. By the time it reaches this place called the synapse, there is something interesting, that these 2 membranes are not connected. There is a gap there. It's called a synaptic cleft. There is a gap, and the gap looks somewhat like this. This is where this electrical signal gets transformed into a chemical signal. And then the chemical signal again leads to an electric current in the next neuron, and then this process continues. This is very important for us because this has been thought to be a very special property of neurons. The fact that neurons can communicate through both electrical and chemical signals. So we will come to this later, that square. Don't worry about it too much. So how do we study intelligence? I will take the example of learning and memory, but many other processes look the same. This person everybody knows, Ivan Pavlov. Pavlov, what did Pavlov do? Dogs. Dogs are associated with Pavlov very commonly. Why so? What did Pavlov do? Conditioning. Conditioning. Rings a bell? Bell. So Pavlov. Okay. So Pavlov was a physiologist, and what Pavlov saw was that Pavlov's dogs would salivate every time they were shown meat. That makes sense. A dog likes to eat meat. You show it meat, it will start salivating. That response is natural. So what Pavlov did was, before he gave the dog the meat, he rang a bell. And he did this many, many times. And there came a point when ringing of the bell was enough to make the dog salivate. Are you with me? So we call the meat unconditioned stimulus, something that evokes a response naturally. So the salivation of the dog is an unconditioned response, but the bell is called a conditioned stimulus. The bell meant nothing to the dog till we connected it to this meat. Are you with me? Fantastic. And the salivation of the dog then becomes a conditioned response to the bell. Once again, you give dog meat, dog salivates. Now what we do is, before you give dog meat, you ring a bell. The dog learns to associate the ringing of the bell with giving of the meat. Right? So every time the bell is rang, even if there is no meat, the dog will salivate. This is the first kind of learning that we could formally study, which is called associative learning. Associative learning involves the association of two stimuli. Make sense? Okay. So this was at the level of a whole dog. Now people have studied learning at the level of neurons as well. So imagine that this is a system of neurons, two neurons that are connected to each other, where if you activate this one also gets activated. Make sense? Many, many such neurons are present in our body. So the connection, the strength of the connection between these two neurons is called the weight. This term also comes in artificial intelligence literature a lot, which is the weight of connection between two nodes. So this is the weight, the connection between these two neurons. And what people have found is that you can take glass tubes, very thin glass tubes, and then you can poke them into the neuron. And inside these glass tubes, you can have a solution, a conducting solution, and a wire. And then you can artificially give the neuron electric currents. Does that make sense? You have the cell, you take a glass tube. This is a very thin glass tube. Inside this glass tube, there is a conducting solution and there is a wire. This is connected to a machine, and using that machine, you can artificially give currents to the neuron, which will either activate the neuron or deactivate it. Makes sense? So what somebody did is that they put one electrode here, another electrode here, and they activated both of them together. Same process. Wire inside, wire, you can give current, and the current activates the two neurons. And what they saw is that the number of times you simultaneously activate these two neurons, you increase the strength of their connection. This is called, this was famously captured by somebody called Donald Hebb in a sentence that says, neurons that fire together, wire together. This is called Hebbian learning. Again, it's fundamental to artificial intelligence. It's called the Hebb's rule or Hebbian learning. So what happens in Hebbian learning? I will try my best to take you through this, but at any point of time that you think I'm not doing good enough a job, please stop me and I'll try to do it again. This is the top of this neuron. Okay, the second neuron, let's zoom into this part, and you have something like this. Okay, it's getting these green dots, which is what are these chemicals called neurotransmitters. They help carry a chemical signal from one neuron to the next. Makes sense? What these neurotransmitters do is they connect to proteins on the membrane of this cell. And some of these proteins are ion channels, which means if they open, they let certain ions flow through them. Many of them are calcium ion channels. So you have neurotransmitters come, they bind to these ion channels, they open them up, calcium flows in. Calcium can do many things inside a cell, but one of the things that calcium does is it activates a protein, another protein, called protein kinase A. Kinases are enzymes that can phosphorylate other things, which means they take a phosphate group and they attach it to something else. Okay. So calcium activates protein kinase A. Kinase, it's something that can go and put a phosphate group on another protein. And what this protein does is it goes and puts a phosphate group on a protein called Krea. Krea is present in the nucleus of the neuron. It is not present anywhere else. Makes sense? Okay. And once Krea is active, it can go and bind to DNA and produce more proteins. These proteins would be used to expand this connection. Yes.
How does the calcium reach there?
So calcium is there because what the fluid here has all the ions. It has sodium, it has potassium, it has calcium, it has chlorine. We are consuming food, and from there, the calcium is also here. Now, the thing is, calcium is usually outside the cell, and within the cell, it is trapped in specific organelles, like mitochondria, for example, has a lot of calcium. When these neurotransmitters bind to these ion channels, they open these ion channels up, and then calcium can flow in because the concentration of calcium is very high outside the cell. So calcium rapidly flows in, and once calcium rapidly flows in, one of the things it does is it activates protein kinase A. And protein kinase A can then activate something in the nucleus called Krea. Now, Krea is what we call a transcription factor. A transcription factor is a protein that can bind to certain sites on DNA. And once it binds to those sites on DNA, it can produce mRNA. mRNA will be used to produce more proteins. And these proteins will be used to expand this synapse. Yeah, area-wise, volume-wise, number-wise. So we would have more boutons come up. The length of these boutons, the area of these boutons will increase. The number of receptors would increase. Yes. From where? Yeah, so pKa, once it is active, it can move to the nucleus. It usually can't move to the nucleus. It can move to the nucleus. Once it goes to the nucleus, it activates a protein called Krea. Krea is a transcription factor, so it can bind to certain sites on DNA and produce mRNA. Once there is more mRNA, it can be used to produce more proteins. And these proteins are going to be used to expand the synapse, like build the whole synapse large. So you would have more of these boutons come up. So let's go back. So number of these boutons are going to increase. Their surface area is going to increase. And the number of receptors is also going to increase. All of this will be synthesized from the proteins that are being produced here. Does that help?
Is there an upper cap to this expansion? Because it seems like a chain reaction.
Yeah, there is an upper cap to this because what people have seen is once the calcium has entered, it's also rapidly sequestered. So all that activation has happened. And then very soon there are organelles inside the cell, like for example, mitochondria or endoplasmic reticulum that will take in the calcium and store it so that it can't keep doing this again and again. The other thing that people have seen is that if you keep triggering this neuron to build its synapse, there is a time when it plateaus. Physically, it's not possible. There's a limit to the number of expansion, how much you can expand that synapse. And you keep doing it after a point of time, the synapses start dying. That also happens. Does that work, Vivek? Oh, why is Krea there is a great question. We don't know. There are many things inside the nucleus. Krea is one of them. Why is Krea there? I really don't know. It's just there. It's one of the nuclear proteins. It's not found in the cytoplasm. Usually it's only present in the nucleus, and it acts as a transcription factor when activated. Yeah. Okay, this is important. It will come back to us in a while. And it's okay if you don't understand every single bit of this, but as long as you can remember pKa, Krea, we should be good. Does that work? So we know that pKa is a kinase. Kinase means it phosphorylates. So it phosphorylates this thing called Krea. Krea goes and binds to specific sites on DNA. They are actually called CRE. So Krea stands for CRE binding protein. So CRE is a C response element, which is a part of the DNA, which is where Krea binds. Once it binds there, it can lead to an increase in protein production, whatever is under that site. Yeah. Make sense to everyone? Any questions thus far? Yes.
There's an upper limit to the stimulus, and you mentioned that there is
Yeah.
time to a stimulus would also be limited accordingly.
So the response, so in terms of plasticity, in terms of the learning you are saying, or the response here? So like inflow of calcium level response or production of proteins and building of the synapse response? Yeah.
Deficient in calcium, right?
Yeah,
Stimulus is automatically reduced.
absolutely.
So the response has
Absolutely. Absolutely.
either the reaction, which is
Yeah. So these are actually very fast processes. I mean, calcium sequestration, for example, is a rapid process. Production of proteins is slightly longer, but the way production of proteins works is that you have a mRNA that is produced here, and then that mRNA is shuttled out of the nucleus, and then things called ribosomes sit on the mRNA. And because we are eukaryotes, which means we have cells with nucleus, multiple RNAs can sit on the same protein site, so mRNA, and keep making more proteins. So there is a rapid turnover of protein, and after some time, the RNA is degraded. So there are many, even if it looks like there are many steps, all of these steps happen quite fast. The point is, once these proteins are built, it takes some time to build the synapse. I don't know how long exactly that is. That's not.
Everybody have taking the calcium, protein, carbohydrates. But why the various level of intelligence differ from person to person?
Yeah, I think that is because we are talking about different levels of different definitions of intelligence. So what we are talking about perhaps is intelligence as we understand more commonsensically, that, oh, you are not very smart kind of intelligence. At least biologists don't study intelligence that way. So I'm going to show you what we mean by intelligence in a while, and perhaps we can come back to your question. Does that work? Okay. Any more questions on this? I'm happy to answer them. Okay. Great. So what we understand at the end of this is that studying intelligence can happen at a behavioral scale. So there is the entire animal, and the animal behaves, and you can make a comment about intelligence like learning and memory at the scale of the animal. You're studying behavior. Does that work? And you can study it at the neuronal scale, which is you can look at the neuron and you can see what is happening between neurons. And if you want to go deeper, you can look at it in the molecular scale, which is you peer deep inside the neuron and see what is happening at the molecular scale. Does that work? Okay. So here is what I thought we could do, is we could throw out the neuron from the business and see where that takes us. So I'm going to primarily be talking about animals that don't have brains and in fact don't have neurons at all. So the first study that I'll be talking about is very recent. This is also a November 2024 study, where this is what the researchers did. They took a neuroblastoma cell line. Neuroblastoma is a kind of cancer that happens in the adrenal glands. Adrenal glands produce hormones and they are located just above the kidneys. So ad is close, renal is kidney, so adrenal gland. And this cancer happens usually in children, very young children below 5 years of age. And what you can do is you can take out some cells from the tumor and you can immortalize it in the lab. You can culture those cells in the laboratory over multiple generations to the point where these cells, where the cells do not need the entire body to grow. Okay? So you can immortalize cells in the laboratory. So these are called immortalized cells or cell lines. Okay? You can do that. You can take some cells out from the body, you treat it with a series of chemicals, and you provide it with all the nutrients it needs from outside, and the cell can keep growing. That is how you make a cell line. So what they did is they took two such cell lines, one that was derived from a neuroblastoma. It is a cancer of the adrenal glands, and the human embryonic kidney. So this cell line was made, perhaps some 100 or 200 years back from an embryo. From its kidney, a couple of cells were taken out, and those cells were immortalized in the laboratory. So these cells can keep growing in the lab, as long as you're providing it with nutrients from outside. So notice that none of them are neuronal cell lines. Neuroblastoma is immature neuronal progenitors. Human embryonic kidney is kidney cells. And what the researchers did is that they added activators of pKa. You remember pKa from the previous figure? pKa is what is being activated by calcium in neurons. Are you following me? Now, because these cells are not neurons, you can't activate them by opening ion channels. So you have to add a chemical that activates pKa. It's an artificial activation of protein kinase A in non-neuronal cells. And then what they did is they measured the expression of a fluorescent protein that had been attached to the part of DNA that is transcribed by Krea. Once again, they added artificial activators of pKa, and they wanted to see whether this pathway, which is indicative of learning, this is a hallmark of learning, the activation of this pathway, whether this pathway can be activated. How do you know Krea is activated? The best way is to check whether mRNA and protein production is happening. So you can add a DNA sequence using genetic techniques. You can add a DNA sequence downstream or just where Krea is supposed to start producing the mRNA. And if your experiment works, the addition of pKa activators should lead to production of that reporter. The fluorescent proteins are not found in the cell. It has to be added artificially from outside. So as long as you see the fluorescent protein, we should be able to say that these cells are showing one hallmark of learning, which is the activation of the pKa-Krea pathway. Make sense? Great. So this is a Nature Communications paper from just a couple of weeks back, and something that I'm writing about currently. So what they did was something very interesting. So the activator of the chemical of the pKa, the chemical activator of pKa, is dissolved in some compound. It can be water, it can be ethanol. So to ensure that the effect that they see is not because of that compound, they have to use, because of that solvent, they have to use what is called a vehicle control. So let's say compound X activates pKa. That compound X has to be dissolved in something and added to the cell. How do we know that it is not that solvent that has led to the effect that we see? So to a different group of cells, you add just the solvent without the compound. That is called a vehicle control. With me? Okay. Then you give one pulse of the activator. You give it for a couple of seconds, and then you take it away. Then you give many pulses together and take it away. So this is called a masked training. And then what they did is they did what is called spaced training. Spaced training means that you give one pulse, wait for 10 seconds, give another pulse, wait for 10 seconds, give another pulse, wait for 10 seconds, give another pulse, wait for a longer duration, and then do this thing 4 times. Make sense? It's like you have a electrode into the neuron, you give one electricity, that would be masked activation of the neuron, or use a blip, 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 wait it out. Then you do blip, 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 wait it out, blip, 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 blip. blip, 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 blip. That is called spaced activation. Then you can also do then they did a different condition of the spaced activation where each blip had a 20-second gap between them. And then there was another stage where they had 30-second gap between them. Each of it was repeated 4 times. So they are basically working with 6 groups of the same cell, one that has no compound, one that has the compound added only once, another that has a lot of compound added together, another where the compound is being added in multiple blips, each spaced 10 seconds apart, and this whole thing is happening 4 times. And then there's a 20-second and there is a 30-second criteria. With me thus far with the protocol of the experiment. What they see is compared to one single activation of pKa versus the masked activation, where you add a lot of compound at the same time, you actually see higher activation of pKa and the Krebs pathway when you do blip, 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 blip into 4. So the first spaced condition of activating the cell leads to highest activation of the pKa Krebs pathway. Yeah. This tells us 2 things. One is that a hallmark of learning is seen in cells that are not neurons. These are kidney cells and cells derived from the adrenal gland. Both of them show activation. This is the 0. Both of them show activation of the pKa Krebs pathway, but they also show another hallmark of human learning or mammalian learning, which is the masked space learning effect. Which means that if you train in short pulses over multiple durations, instead of training yourself in a large pulse at one time, learning is more effective. One way of thinking about it is, instead of studying for 8 hours continuously, it is easier for us to learn something if we study for 30 minutes, take a 10-minute break, 30 minutes, take a 10-minute break, 30 minutes, take a 10-minute break, 30 minutes, 10-minute break, maybe a 30-minute break, and then start that cycle again. Learning is more effective in spaced conditions as compared to masked conditions. And our immortalized kidney cells and immortalized adrenal gland cells are showing the masked space learning effect, which is really huge. This is something that was seen as the hallmark of neuronal learning. And now we know that this is no longer the hallmark of neuronal learning. Yes.
Why is the pKa to Krebs pathway considered learning?
Because this calcium-mediated activation of pKa happens predominantly in neurons.
Are you saying that pKa is not present in others?
pKa is present in other cells, but the activation of pKa through calcium and its resultant activation of Krebs, and then the production of mRNA and protein that builds the synapse is something that happens only in neurons, or at least has been reported only in neurons.
Can kidney also build synapses?
We don't know. We don't know yet. I think we need more research to understand what it means at the phenomenological level, what it means for the kidney cell. Remember that this is an artificial activation, so the compound is not present in our kidneys. So yeah, we don't know. We have to study it in an in vivo system where you have not taken the cells out. But for now, what we see that at least these cells have the potential to show something that for a very long time has been thought of as a hallmark of neuronal learning. Lucifer is the reporter. You can read about it. Yeah. Everybody understands this experiment. What it shows is kidney cells and neuroblastoma cells. I'll come to you. Kidney cells and neuroblastoma cells show masked space learning effect, which is a hallmark of neuronal learning. Yes.
This shows that it's a process of growth and not learning. Right?
Yes, you are right. Unfortunately, or fortunately, I don't know, a lot of biologists who study learning at the molecular and the neuronal level see this as a readout of learning. If you are talking to somebody who does behavioral biology, they would use something like Pavlov's models to study learning. But a lot of neurobiologists who look at neurons would see the growth of synapses as an indicator of learning. So we are talking in terms of interpreting what we are seeing under the microscope as a growth of synapses. Absolutely. But there are neat experiments that correlate very well this growth of learning into behavior of animals. So in sea slugs, this person called Eric Kandel has done experiments and won a Nobel Prize, where he showed that whatever you do at the animal level, so the animal has a behavior like Pavlov's dog that you can trigger. You can also mimic the same at the neuronal level. So in the same animal, you can dissect, you can keep the animal alive, reveal a series of neurons, and you can stimulate them and study the behavior simultaneously. So those models have been very helpful in telling us that what you see as growth at the neuronal level appears as learning at the animal level. Yeah. Okay, this is cool, but this is the star of the show. This is yes, it's a protist called Physarum polycepharum. People think that it is a fungus, but that's a miscategorization. It used to be categorized as a fungus in the past. It's a slime mold. You see it in dark, wet tropical forests. It's yellow in color. It's all over the forest floor. Sometimes it's under the leaves. Sometimes it's on the bark. It belongs to a group called Protista, which many people think contains everything that biologists can't explain. So there is this curious organism. It's very interesting. So you see this big blob. So this is grown in the laboratory. This is a Petri plate. It's growing on agar. I think all of you might know what is agar. It's a jelly-like substance that you can grow things on. So there is this. This is where you inoculate the organism. You put a blob of it. This organism is basically a cell membrane with many nuclei in it. So we call it a acellular organism in some sense. So all these branches are part of the same cell. So what this organism does is that it spreads out a fan of its body, and that is called a pseudopodia, a false leg. It senses something and then moves a little further. So it grows another branch out. Then that branch senses something, then it grows another branch out. It's a very, very curious organism. This photo is courtesy the Weiss Institute, Michael Levin's laboratory. So you see these fans are pseudopodia, these fans out here. So it started somewhere here. You have a fan, move forward, fan, then a branch emerges, fan, branch emerges. That's how the animal navigates its environment. I will actually show you a video if it works toward a little later. But this animal did something remarkable in 2000. So what these researchers did is that they took, this is the Petri plate where this animal was supposed to be grown, and they put plastic sheets in a manner that it created a maze. They created a labyrinth that the animal had to navigate. And then they took this animal, they broke it up into many, many pieces, and they sprinkled it all over the plate. With me thus far. And as expected, this thing grew all over. Nothing surprising there. It just grew all over. But then what they did is they introduced agar blocks containing food. This is the maze. Sorry, the image is blotchy because the paper where they reported has a blotchy image, not my fault. But they put these agar blocks which contained food. And if you notice carefully, what this thing did is that it retracted its entire growth. So what was entirely yellow until a little while back, this thing retracted its entire growth and found the shortest distance between the two agar blocks through the maze. Yeah. Yeah, so what happened was so it looks like this, right? So let's say you want to inoculate this animal in a different Petri plate. Inoculate is introduce this animal in a different Petri plate. You will take some chunk of this animal from here. Yeah, this is, like I said, this is an acellular organism. It's basically a single cell with many, many nuclei. So you'll just take a chunk and you would dissolve it very well in water or some medium. And then you would put drops of it in different parts. And as you would observe, even with bacterial or fungal colonies in the laboratory, if you do this, it will grow everywhere. Then what you do is you introduce food, and you introduce food at a start point and an end point of the maze. And what they observed was that all this growth was immediately retracted, and this organism traced the shortest path between the two food blocks. This study won the Ig Nobel Prize in 2000. Do you know what is the Ig Nobel Prize? I know that thanks to TNSF, we all know what is the Nobel Prize and who wins it every year. The Ig Nobel Prize is awarded to studies that first make you laugh and then make you think. And it's actually quite prestigious to get an Ig Nobel. So this study got an Ig Nobel Prize in the year 2000 for demonstrating that slime molds can navigate mazes. They can compute the shortest distance between the start and the end point and retract all their growth to eventually find the shortest distance between these two places. It's one of the first and the most elegant examples of intelligent behavior in brainless or neuronless organisms that we have. And the interesting thing is that they didn't stop there. This is what the person wrote in their paper. This was published in Nature. It's a brief communication. The whole paper is one figure and 3 paragraphs. So they basically said, we put up maze, we put this animal grew all over, then we put food, animal retracted everything else and found the shortest path. That is the whole paper. It's just half a page if you want to read it. This remarkable process of cellular computation implies that cellular materials can show a primitive intelligence. They were right in saying that it's intelligence. They were not right in saying it was primitive. And I'll show you why. This is another experiment that was done by a different group in the year 2012. So again, you see this animal, all the things that you've seen before, the branches and the pseudopodia. What this animal does is wherever its branch goes, it leaves a transparent slime. That is why it is called a slime mold. So wherever this animal touches, there is a transparent slime that is formed. And researchers have observed that where there is slime, the animal doesn't go again. So it's almost like there is a spatial memory that the animal is building through the slime outside. I'll come back to the question of spatial memory in a while. But you have a extending pseudopodia here, a pseudopod here, a fan-like structure which the animal is using to sense its surrounding. There is the search front, many pseudopodia at one location that are trying to sense where to go. There is a tubule network, these branches. This is where the culture was put. So you have a blob there. And this is the extracellular slime. And wherever there is slime, there should be, the animal shouldn't be there. The animal avoids a region where it has gone earlier. So here is a very interesting experiment that they did. They took some of this slime, they just picked it up. You can scrape it from the agar. And they applied it all over an agar plate. So everything is slime. You get it? The animal should ideally have nowhere to go. Then they put a block, again, plastic sheets you can put in the agar plate. You put a block. And on this side of the screen, they put a drop of glucose. Like most things in the living world, our slime mold also really likes glucose. So they put a drop of glucose here. And because glucose is here, they said that there would be a gradient of glucose that will spread. This has the highest concentration of glucose, and then there is a gradient of glucose that is spreading. They put the animal here. And they said that if the animal functions optimally, we know that it can find the shortest path. So the animal will take this path to the glucose droplet. Are you following me? There is a reason why it will not take this path, because it needs to bump against something to turn. So they said that this would be the optimal path. So the slime mold will try to come straight, because remember gradient of glucose. So it's going to try and come straight. Then it's going to hit on this wall. So it should turn, hit again, go up, turn, come down. That's the optimal path that the slime mold should take. But remember, there is slime all over. And we said that slime mold does not like to go where slime is. So here is what they saw. That when you do this experiment on blank agar, this is the number of times, or this represents the percentage proportion of times that slime mold is able to find the glucose drop. When there is extracellular slime, that performance drops because it can't navigate. It doesn't like slime. It interferes with space navigating phenomena. Mean time to reach the goal increases substantially when there is slime, because it has become slower. It can't be as fast as it would be if the slime was not there. What is the amount of time that they spent in the trap? The trap was that this is the trap. What is the amount of time that they spent in the trap? Increases substantially, again, because they can't navigate optimally. And then they found the mean time spent on own slime trail, because we said that slime molds do not like to spend time on their slime trail. On blank agar, where it has moved, left a slime trail, it will not spend a lot of time there. It will move forward or retract. Where there is a lot of extracellular slime, it has nowhere to go, so it spends a lot more time on its own slime trail. And this is from Wright et al. It was published in the Proceedings of National Academy of Sciences, USA in 2012. And they said that this is proof that the slime mold is using its slime as externalized spatial memory. And when you take some slime from somewhere and you put it everywhere, it is simply unable, it's interfering with its navigation processes. Now, this is very interesting because we also have spatial memory. We do, which is why we remember that Anna Centenary Library is here, the 5th floor is here, and you have to take the lift on the right to climb up. We also have very solid spatial memories. Rats have great spatial memories. Honeybees have spatial memories, but all of it is encoded in the brain. In human beings and rats, there are specific cells called space cells and grid cells, which encode a map of the world. The slime mold is encoding that world outside. It's an externalized spatial memory. Something that for many neuroscientists was inconceivable because we were too busy looking inside the head. And one of these is a map of the Tokyo rail network. Tokyo is the capital of Japan. And one of these images is the map of the Tokyo rail network. Each of these dots is a station. And one of these is the path that slime molds make if you put food in the same location as you would in a map of Tokyo railway network in Japan. So you understand what I'm saying? You say, okay, there is station 1, station 2, station 3, station 4, station 5. You take this and you put food exactly like that on a Petri plate for the slime mold. What do you notice?
We use the slime mold to solve the problem without a slime.
Yes, one of the things that you see is, can you guess which one is Tokyo Railroad? On the right. How did you know? Huh? It looks longer. It looks longer. There is some guesswork involved. Great. And you're correct. You're correct. This is the growth of slime molds, and that is the Tokyo rail network. But the first thing that you must observe is some eerie similarities. You see this larger structure. You almost see that larger structure appear here. You see this connection. You see that connection out here. You see this connection, you see that connection out here. It's actually eerily similar that a single cell that has been given food can trace a surprisingly accurate map of the Tokyo rail network, but then you do a cost efficiency analysis. So you basically look at the amount of energy that the slime mold is spending to build that path and what is it getting, the amount of food that it's getting. You actually see that the slime mold path is slightly more efficient. It's marginally more efficient, but it is more efficient. And they have done this several times. And this was a science publication. Science is one of the biggest journals in science. And these authors have suggested that if you want to build paths, then slime molds might be a great place to start because they can give you some of the most efficient maps that connect different spots of food put together. And this similarity is very striking. I know that there are some differences. This line is missing here and all that, but it is still remarkably similar for an animal that is a single cell and has and looks very different from us, does everything very differently. And here is I believe the most fun thing. I'll tell you what it is, but let's just enjoy it for a while. Did you see that? It's lovely, right? It started navigating here, then it navigated here, and then it at some moment decided I'm going this side. Let's watch it again. We can watch it again. Let's see if we can pick it up again. So see, it's tracing the top, then it's tracing the bottom, and then it is deciding I'm going down, and it's going down. Do you see anything that is down? Yes. So what is interesting is that there is no food on this plate. There is a heavy glass disc here, and there are 3 heavy glass discs here. No, nothing has food, it's just weight. And it looks like slime mold is sensing the direction of more weight and choosing to go there, deciding to go there. So this finding gets slightly complicated because then what these people did is that they placed these 3 discs on the top of each other, and then this preference vanished. So it's a particular orientation of these 3 glass discs, not just the weight that the slime mold prefers. And they have come up with a really, so they did a computational simulation of this phenomenon, and they came up with a very cool explanation. They said that because the slime mold, I don't know how to show this with my mic. I'll show off my mic for a bit. They said that the slime mold basically makes a movement that looks like this to travel. Yeah. So it drags the surface on which it is traveling for a brief moment. And if there is more weight here, this part is going to give more, it will be harder for the slime mold to drag, right? There will be more resistance, more strain on that part. And the slime mold prefers more strain. Are you following me? Great question, we don't know. Why does the slime mold prefer more strain? At least in what I read of this paper, I didn't see. I'm happy to read it again, but I don't think this is enough for us to speculate on why. But what we do understand is that this is a behavior that is not just a sensation of weight. It is a sensation of strain. And this is a fairly sophisticated response. This is mechanosensation. It's a sensation of physical processes and responding to them. The slime mold is deciding to go to that part of the plate where there is more strain, not just more weight.
Prefer surface area over concentrated. You said you stack the weight, that doesn't.
Yeah. So the thing is, concentration might not be the right word because there is nothing, like there is no food in this. It's just a weight, like a glass slab.
When you stack them on top of it,
Yeah,
it doesn't
so then the preference vanishes. It goes both sides equal number of times. So the preference is to the, so what they found is that how you put the glass discs, not just the weight of it, will determine the strain on the surface. So strain is the change in length when you apply a force divided by the initial length. That's the strain. So the strain, that quantity here is going to vary depending on how you place these glass slabs. And it looks like the slime mold is sensing strain. So, so really, a skill that we simply didn't expect the slime mold to have, that's quite intelligent. So we know that the slime mold uses physical cues for decision making. That is like us deciding to go and read in a part of the library that is well lit over a part of the library that is not so well lit. Yeah, it is. Let's look at it again. I can look at it the whole day. It's quite fascinating, and there are many of these videos in the paper. It's really cool. Yeah. A lovely branch of slime mold. So finally, I want to talk and I want to close by talking about what does us finding more and more interesting things about intelligent behavior in brainless organisms tells us not just about science and humanity. Thank you very much to Dr. Vijayan for finding this image in the poster. I don't know who made the poster, but sorry? Sundar. Thank you very much for the poster. Lovely choice. And I decided to go with it. This is, if you look at the whole idol, it's a Buddha idol without a head. And I thought nothing could be apt for a talk that is titled No Brainer. So, so yeah, lessons. So I think the lessons for science are clear that if we are starting to recognize these animals as intelligent, then studying these animals should tell us a lot more about intelligence than what we understand right now by studying the various animals that we have. And that is good because that means we are not going to keep looking back into our heads. We are going to sort of step out, explore animals that we have seen as just foraging food for the most part, as something that can tell us fundamental things about intelligence. And this is where and once we understand how intelligence works at these levels, it should be possible for us to build intelligence in some sense. And that is what Dr. Vijayan was talking about when he spoke about my recent report, where I reported on this study from a laboratory in the Saha Institute of Nuclear Physics, Calcutta, where they plugged in several genetic sequences into a series of bacteria and organized them in a manner that it resembled artificial neural network type architecture. And then they came up with a pretty cool way of communicating with the bacteria in binary code. So they chose 4 chemicals. The presence or absence of those chemicals could be read as 1 or 0. And once you have a binary code, you can communicate with the bacterial computer. And they saw that that you're able to make this bacterial computer do some interesting math problems. You can ask it whether a given number between 0 to 9 is prime or not, and it can respond in yes or no. You can ask it if a letter between A to L, I believe, is a vowel or not, and it can respond in yes or no. And you can also ask it whether what is the maximum number of pie pieces you can get if you take a pie and cut it only in straight lines. And that is an example of an optimization problem. People find best solution to a particular problem. And looks like that artificial neural network type architecture is also able to achieve that. So I think those implications are quite obvious. I am more interested in what it means for us as human beings to now know that as we are trying to build intelligence that can literally replace God, we are also changing the great chain of being. We're trying to get to the top by saying that we can create intelligence. And perhaps what finding intelligence in brainless, neuronless, single-celled, blobby organisms tells us, reminds us is that climbing up that great chain of being is not the point. Point is to remember that we are part of a very curious ecosystem of living beings, where all these living beings have very cool properties. They're doing very cool things themselves, and that should from the time that I was studying neuroscience to the time that I write about it, recognizing that that we got this belief that we are the most intelligent beings on the earth, we got it wrong, has made me humble in looking at the world. And I hope what this talk instills in you, beyond the wonder for science and fascination for it, which I hope it did, is the same sense of humility that allows us to appreciate these blobby things find their way around in a Petri plate. Yeah, that's all I have to say. Thank you.
Thank you.
Thank you.
Excellent. Before we get into the question and answer session, for Mal, another library, Central Library, we will remember how to honor
Thank you so much. Thank you. Thank you so very much.
Questions? I think the session itself was interactive and many people asked.
Yeah, yeah.
Ask the question.
Yeah.
Any further questions?
Yeah, I'm happy to answer questions. Yeah, there are 2 questions there.
This is sort of
Excuse me, ma'am.
with regards to that.
Let's have this also so that people
Okay.
know.
So that you're also heard on Zoom. Your
Um,
voice must be imprinted on the internet forever. Yeah.
okay. So
Have this-
No, thank you. Thank you.
I remember you spoke a little bit about the spaced repetition part.
Yeah.
You spoke a lot about the spaced repetition part, but does it matter if it's variable? Does it have to be like 10 minutes pause, 10 minutes pause, or can it vary?
So, so what people have done is that they have tried this out with different spaces between the stimuli. And for all organisms, there seems to be the curve rises to a point. So where learning increases when you space out the stimuli.
Mhm.
And then there is a point when it decreases. So there is an optimal point, but that is not same across species or animals. Then it has to be calibrated for each species that you're working with. So it does. So the short answer to your question is it does matter what is the space between these, but it's not universal. So I can't give you a number for that organism. When you start your lab, you do this experiment with different spaces, and there is a time when you are able to calibrate it. You're going to get a sense of when the graph starts to fall, and just before that you should stop.
Thank you.
I think the person right back had a question.
Oh, sorry.
Yeah. Oh, sorry.
So you explained quite about biocomputers. So can you explain brief about it?
Okay, yes, I can try. So yeah. So the idea is that can you get bacteria to grow to do the same things that some of our computers do? And when I say, I mean, don't think that bacteria are going to do simulations of the world. But yeah, but in the sense that can we give it problems and can it solve those problems, abstract problems that it can solve? And that is seen as important because bacteria are very easy to grow. They require very few resources. So in compared to silicon-based computers, which are extremely resource heavy, environmentally damaging, bacteria are perhaps a more sustainable alternative if we ever get to the point where biocomputing at that scale is possible. But the idea is this, that you take a bacteria, and we know this particular bacteria called E. coli, its genome extremely well. And we have tools to pretty much cut out and put in whatever we want to put in its DNA. So you put in a couple of DNA sequences that can sense the presence or absence of some chemicals. And you put in these sequences in a manner that each bacteria acts like a Lego block. So you have bacteria type A that you've synthesized. When combined with bacteria type C and bacteria type E, it should be able to build an adder. So something that can add numbers. You can then, if let's say bacteria A, bacteria B, and bacteria C, 3 types, when put in a solution, should be able to build what is called a subtractor. And you do these experiments to the point where you see a artificial neural network type architecture emerging. What that means is when you start plotting its input-output function, you know that a neural network has a very specific input-output function. It's called an activation function. And activation function looks pretty much the same for all neural networks. And it looks the same for this bacteria as well. At that point, you can say that you have created a single layer artificial neural network. and you ask it questions. You ask it questions by by regulating the presence or absence of 4 chemicals. So that gives you 4 binary bits code your question in, and you use those bits to code the question, ask the question to the bacteria, and the bacteria has fluorescent proteins that it can express or cannot express, which is again 1 and 0. So it can also respond back to you in a binary. You want more bits, you can add more fluorescent proteins. So presence or absence of 4 fluorescent proteins gives you 4 bits of responses. So that's largely where at least the study that I covered stands. It's actually published in a very respected journal, Nature Chemical Biology, where they built a system like this and they made it do computation of a prime, basically identifying whether a number is a prime or not, whether a number is a vowel or not. I don't know if, there is a mathematician in the room for sure, but mathematicians work with things called Haar graphs. I don't know exactly what they are, can't try to explain it, but you could also ask this bacteria if if you give it a number and the number is supposed to generate a connected or Haar graph or not, and the bacteria can respond to that. It can come up with it can compute the optimization problem of pie slices, which are all fairly complex abstract computations to do. So that's quite cool. Does that answer your question? So, yeah.
I think there are,
Oh, there is a question there. Yes.
I hope there will be more experiments on exploring the intelligence, not only in the human being, also in other things like vegetable kingdom and mineral kingdom and all.
Right.
There may be other kind of experiments on exploring intelligence in the mineral kingdom. There is no dead matter in the world.
Right.
That has to be also explored. It is good to investigate how the intelligence is operating in such a brainless organism through certain mechanism. That is what we require.
Yeah.
We want a microscope and other things to see that, to observe how it is happening, how the intelligence is spread over different places without any hardware.
Yeah.
It's a wonder.
Yeah.
So we have to explore further and see that how it is evolved further.
Yes.
Because there is no hardware previously, and then hardware comes in the human being. How it is evolved like that, how it is evolved, and then the intelligence become more complicated.
Right.
The Hebb theory says the interconnectivity among all the neurons that depends upon the activity we undertake.
Right.
Certain activities bring more areas in the brain, connection, strengthen and all. More learning takes place. So it has application even in the educational field. We have to bring more areas, integration. We call it sensory integration. That will promote learning and intelligence.
Right.
This is the Hebb theory has the implication. I don't know how that theory has applied in this area, brainless organism. How it is applied? This is my question. Can we apply the same thing?
Yeah.
But strengthening is important. Strengthening here strengthens.
Yeah.
But in this case of brainless organism also, that is strengthened gradually.
Right.
That is what my remarks. Thank you for a good experiment.
No, thank you. Thank you. Your question is very valid. So to answer the first part, so I don't know what's happening with minerals, but certainly with plants, there are some labs that are investigating intelligence in plants, particularly carnivorous plants. So you have Venus flytrap and pitcher plant, which grow in very poor nutrient soil. So they rely on trapping insects and digesting them for their survival. And one recent experiment has found that so every Venus flytrap has a trap It's a leaf that has spines, and then a fly sits on it, it closes and traps the animal, digestive juices are secreted, the animal is digested. And they have found that so this fly trap, the leaf of it has hair that senses movement. They're mechanosensitive hair. And what they have found is that using that movement, the Venus flytrap actually keeps a count of the number of triggers. So it counts 2 triggers before it closes the trap. It counts 3 triggers before it starts secreting the digestive juices. So there is some rudimentary or primitive, and I'm very scared of using the word rudimentary or primitive because we called slime molds' intelligence primitive and they mapped out Tokyo for us. So there seems to be some ability to count in Venus flytraps, and counting is very, yeah, I mean, it's observed in complex animals. So dolphins can count and human beings can count and so on. So with plants, I think there is substantial interest in investigating intelligence. The second question is whether models of neuronal learning apply to these, especially things like Hebb's rule and all. Frankly, I don't know, but my sense is that it would be somewhat useless to try and see this as an extension of what we understand about human learning or mammalian learning or animal learning. The idea is that having access to learning that we have never before seen should give us something new about learning itself. And I'm hoping that people will try to find newer models of learning from slime molds instead of trying and modeling these slime molds as a form of neuronal learning. But that's I'm not a practicing neuroscientist anymore, but that's my sense as somebody who's reading up on this. Yes.
There is some theory which says that the central nervous system in the human body has an electromagnetic force by which the extrasensory perception is being created.
Okay.
This ESP available in human being, it's available with animal also. For example, there's a plant, Tamil, it's called the total cynic,
Okay.
Touch me now. If you
Okay, yeah.
touch it, you will kind of
Yeah,
feel it.
absolutely.
And bats are just flying, they're calculating the distance
Yeah,
by the vibration of the wings.
absolutely.
Bats.
Yeah,
So this ESP is available with animal also.
right.
What's the difference? Can you tell me, sir?
Difference between what?
The ESP, that is due to the electromagnetic force in
Right,
the nervous system of the
right,
human being. The
right.
same faculty available in animal also.
Yeah,
There.
yeah. That also has the same nervous system, huh?
Yes, exactly.
Well, very identical, huh?
But what's the difference? That ESP, what's the difference? Why?
Okay.
In that way, the animals are more intelligent than man. I mean,
Okay. That.
or she.
Yeah, right. I get the question. No, I think that is the thing that we can't use one scale of intelligence to compare human beings and animals. So we can't say that a bat has a sensory quality that we don't, which makes the bat more intelligent than us. All we know is that the bat is different. And this is a question that was asked by this person, Thomas Nagel, a philosopher of science, who asked the question, what is it to be a bat? Nagel asked that given that a bat maps its world through sound, not through vision like we do, can we ever imagine what it is to be a bat? And Nagel seems to land at an answer that says no. And that's been contested by others. But yeah, the idea is this, that we can't use the same scale of intelligence to compare animals and human beings. In fact, we can't use the same scale of intelligence to compare different human beings. Can't use the same scale of intelligence to compare different cultures. So, for example, I have a friend who studies how street vendors in the cities of Hyderabad and Mumbai divide, basically, right? So you go to a street vendor and you give that person difficult calculation for like 37. Can you divide the number 37 rupees into 4 people equally? And I will take a hell lot of time to do that. It's very hard for me, but they come up with quick answers very fast. But they have not been trained in mathematics. Many of them have not completed large schooling, so it looks like some mathematics is embedded and embodied into cultures. And we can't say you are more intelligent or you're less intelligent because you can do this and you can't do that. I do want you to remember that all the conversation that we have about intelligence is not to bolster this idea that there are more intelligent human beings and there are less intelligent human beings. The point was to say that what we think is human intelligence seems to come back in very unlikely places, and that should, that should tell us that there is nothing particular about human intelligence, perhaps. Regarding the touch me not example is very good because that is one of the solid examples that we have of plants learning. There is something called habituation learning, which is that you keep giving the same stimulus too many times, and then the animal or the plant stops responding. That's called habituation. It's like I don't know, you are in a very cold AC room and you feel shivers for some time, then your body adjusts and you stop responding to that stimulus. You can also do that in animals. Like there is this organism that doesn't like to be shaken. So you shake it, it curls up in a ball. But after some time you keep shaking it, it realizes that the shake is harmless. It learns and it just sort of remains fanned out. So touch me not also that happens. If you keep touching the same leaf of touch me not, it realizes after a point, it gets habituated to the touch and it stops closing its leaves. So it's one of the solid examples that we have of plants demonstrating one form of learning, which is habituation learning. It's quite cool. Yeah, thank you. Yeah.
Yeah, so thank you for the talk. You demonstrated that slime molds without any in the absence of neurological processes does show learning.
Yeah.
Does that level of plasticity come with multicellular organisms also? So where does this question come from? I have a kid with ACC. Despite that, the kid can smell. So in the absence of olfactory nerves, where is the smelling coming from, right? So
yeah. So my honest no, it's okay. My honest answer is that I don't know. What we do understand is that the brain is quite plastic, especially in younger children where neurons are not as hardwired as ours are. So there are cases where substantial parts of the brain have been found missing at a much later stage of the human being's life, and the person could continue living a more or less normal life because other parts of the brain took over that function. The human brain is quite plastic. Regarding whether that level of plasticity is found in human beings or not, I don't know. But also that is a question that I don't think can be answered because then we are saying that we have to develop a scale from there and then apply it to human beings. And I'm trying to avoid that kind of comparison. Either take human measures and apply it to slime molds, or take slime mold measures and apply it to human beings. I am learning to see this intelligence as its own kind of intelligence. And tomorrow we discover intelligence in plants. I'm happy living with that as its own kind of intelligence. And I'm glad that my intelligence is one of these many kinds of intelligence.
There was one paper where the slime mold was showing material behavior. It was responding to some kind of material exchange. So,
Okay.
somebody mentioned minerals.
Hmm.
So you have crystals that grow.
Uh,
You have crystals that grow, and they also can probably chart paths
Okay.
by sitting somewhere.
Okay.
They grow apparently.
Yeah.
So is that phenomena known to be different from this phenomena of first line boards?
Oh, I don't know about the minerals, but I think one of the reasons we believe for minerals, at least from what I understand, this might be a physical phenomena at this point. This growth, there are certain forces and conditions that lead minerals to grow the way they do, crystals to grow the way they do. I think for slime molds, this entire idea of scanning, retracting, then at one point stop deciding to not extend the pseudopodia further and then branching out straight. That tells us that it's not a purely physical phenomena. I'm not saying that the sensation of physical forces is not involved, but there is some biology and some life that's happening there. I don't think it's just a physical phenomena, which is what actually for some things like bacteria and all, people have always said that whatever we see as intelligent behavior is just a product of physics. And I think we are increasingly confronting data that disproves that. Slime mold being one such great example where we can't explain this idea of scanning and deciding as a question of physics. We can model it perhaps and simulate it through physical and mathematical equations, which is very helpful to study its behavior. But the behavior itself is perhaps not purely physical.
So, in order to measure the level of the intelligence of the human being, there is IQ, intelligence quotient, and spiritual quotient and emotional quotient. Any formula for this, only integrating these quotient, once intelligence is being measured, human intelligence is being measured. Can you, if you could tell me the formula of this?
Yeah. So I'm not an expert on human intelligence, but what I do know is that IQ tests are very faulty. They have been largely debunked. At least so what is handed out to kids as IQ tests has been largely debunked. I don't think those IQ tests do not capture intelligence. People know that this is some myth that came up in colonial science and lasted for a very long time, but the IQ test doesn't mean anything for human intelligence. Yeah.
Right. If there is no question, and we'll go over with the winding of the session. The last part of the session is vote of thanks by Chellakani. Yes.
Such a fabulous session today. Thank you. Thank you, Sayantan. Such a young age. In fact, we very simply used to scold our kids that, oh, no brain at all. Not even say that, right? Even any organism has such a brain, right? I mean, intelligence. So very beautifully explained how nature, from the theory, from the philosophy, to how the modern technology it integrates everything, right? Wow. It's like how you like and love physics, how you love the science, right? And your area of expertise. So such a marvelous session. So we're really great. And Tamil Nadu Science Forum and Dana Central Library, thank you for your efforts to come here and deliver this beautiful session. Thank you so much. And thank you for all your patience and very good audience today. You guys have a lot of questions to ask Professor Sayantan. So hopefully you can have offline also sometime he can spend with us. So thank you so much. And thank you so much, Dana Central Library.
Thank you.
So Guru for your time and place for us. So thank you. It's a great session today. So let's meet in the next session for us.
Thank you.